Good afternoon, everyone. Welcome to our MHRC seminar this afternoon. It's a real pleasure to introduce Dr. Stephen Alway to our seminar this afternoon. Dr. Alway is currently the Dean of the College of Health-Related Professions and the Professor of Physiology and Physical Therapy at the University of Tennessee Health Science Center in Memphis, Tennessee. He's been there 3 years. But before that, he spent 18 years at the University of West Virginia as professor and chair of exercise physiology. And before that, he went to he was at a large number of other places, including University of South Florida, Ohio State University, Oral Roberts University, UT Southwestern. And through those travels, he's been funded by NIH for more than 28 years. Remarkable. And it's nice to know that he did his Bachelor of Science degree at the University of Waterloo and his master's and PhD at McMaster University in Hamilton. And I remember seeing his first papers come out with people, co-authors like McDougall and Howie Green and Rich Hewson, pioneering papers on muscle and resistance exercise. He's always been involved in studying muscle, aging, apoptosis, sarcopenia, and exercise. So he's a great fit for our seminar series. And today, I'm going to talk about the role of SIRT-T1 in muscle atrophy and repair. Is it a miracle or is it just a mystery? So Dr. Alway, please, if you would, turn on your video and let's share some screens together and take it away.
Must be sharing the wrong one.
That one looked all right.
Yeah, it did. Maybe I'll just flip it up on this other one here. Okay. I guess we're back.
Just make it bigger if you can, Steve. That'd be good.
Is it not filling your whole screen?
Not yet.
Okay, it should have, because it did when we did the try run.
Yep.
Let me drop off, and it's either that one or that one. Now, is that filling the full screen?
It is not.
It is not.
Hit swap displays. Hit swap displays at the top left. That's good. We're good.
Oh, so it's flipped it from what now it's on my laptop instead of my big one, but we'll make it work. So, well, thank you, Dr. Hood. It's really a privilege to be able to remotely cross the border, because with COVID, of course, everything's been shut down for some time. I've known David for a long time and I really have appreciated him as a person and as a scientist. So I really value the work that he's been doing through the Muscle Health Research Center, and we're really delighted to be part of that discussion today. Just to orientate you, when I was at West Virginia University, as Dr. Hood said, for 18 years, I gave a talk one time when Albert Goldberg was at the same meeting, and I mentioned that I was in the School of Medicine, and he got up and asked a question. He said, I didn't even know West Virginia had a School of Medicine. So after that point in time, I started showing people in talks that actually we had buildings in West Virginia, and there was actually some science that could go on there. Well, Memphis probably is an area where not everybody has been, and maybe you have, but this is our Mississippi River, and we're just a few blocks away from that in our medical district where the Health Sciences Center is. This is the backdrop that I'll be using today, which are just some of the buildings that we have on the campus. This is a medical campus. We've got colleges of nursing, dentistry, pharmacy, and medicine in our college, which is the College of Health Professions. And our college actually spreads from the corner of Memphis that you see here right next to Arkansas and Mississippi, all the way to Knoxville, where we've got one department of audiology and speech pathology. And in the campus that we have now here, we have 3 other departments, and we've developed a division of rehabilitation sciences, which is led by Dr. Jim Carson. And in that, we have a Center for Muscle Metabolism and Neuropathology with a doctorate in rehabilitation sciences and graduate students working in the areas of sarcopenia, disuse, aging, cachexia, and then movement disorders. So we're trying to really build up and recruit, strong faculty to our college to help build some of these areas in muscle. So we share a common theme, I think, in terms of our muscle-centric interests. The talk that I want to give you is really a culmination of more than a decade of investigations in the area of sarcopenia, but it really comes back to the question about muscle loss, muscle wasting, and that related to aging and the issues surrounding that. So I just wanted to frame really quickly why we're interested in that and where this might be going. So the underlying question when I began my career about 110 years ago now, it seems, is why does aging induce muscle loss and lead to reduced muscle repair? As I'm sure you all know, or everyone on this Zoom at least understands, is that skeletal muscle, as we age, has a loss. The loss is both in terms of the size of the existing muscle fibers and the number of fibers that exist in that muscle. So an older person just has fewer fibers, and those are smaller and weaker fibers. The process of muscle loss, it seems to accelerate after the age of 50. And if you are in your 80s, it wouldn't be unusual to expect up to a 50% loss of muscle mass as compared to when you were 20. So it really has a lot of functional implications. It's also exacerbated by issues related to disuse, bed rest, stroke, anything that's going to result in atrophy. So what are the bad things that happen in addition to that? Frailty is one of the complications, incomplete muscle repair, which we're very interested in, increased fibrosis in the muscle, the fact that the muscle can't generate as much force related to the presumably not only the number of cross bridges, but force per cross bridge, and there's data to suggest both of those, leading to falls, disability, certainly quality of life independence, and something that's attracted our interest in the past little while has been the increased susceptibility to metabolic disturbances like type 2 diabetes and other metabolic diseases as a result of having less muscle mass that would act as a glucose sink in this environment. In the US, it's a fairly costly health issue as well, exceeding $18 billion. And it's something that's not going to go away really quickly because our population, as you know, is aging. This is a CT scan that you probably have seen over the years, where we would find differences in the size of the muscles. This is a cross-section through the femur. And of a younger person and an older person, you see not only mass changes in the muscle, but also obviously adipose tissue changes. Our lab has been really interested in 2 pieces to this puzzle. And one is the muscle stem cell or satellite cell, and that involvement in muscle turnover, muscle regeneration, potentially some aspects of muscle hypertrophy as well, and mitochondria. The mitochondria pieces that we're interested in is not only the ATP generating components of this organelle, which is clearly an important aspect, but also the signaling that mitochondria can regulate or partially regulate through being healthy or less healthy, and all the other pieces that go into anabolism and catabolism as well. Starting with the sirtuin-1 story, which will be for this talk this afternoon, our interests were does the muscle stem cell and mitochondria become involved in an aging or muscle loss, muscle atrophy situation, and what's going to cause those dysfunctions? Well, one of the obvious things that might be there would be that of reactive oxygen species. And just the fact that we have the opportunity for ROS, for reactive oxygen species of whatever sort, to induce structural and functional damage to these tissues or these organelles, et cetera, that can impact the tissue health is really going to be sort of paramount underlying the mechanisms. So really the sort of framing this whole piece is how do ROS interact with this whole system? How do mitochondria and muscle stem cells become affected with aging, where there's increases in ROS or other situations? And can we slow or prevent these changes in muscle stem cells and mitochondria that might impact the size of muscles in aging or disuse situations? So the story of SIRT1 is certainly not new. As I said, we had studied it for more than a decade, and we've kind of changed some of the focuses in our lab more recently. But the interest that we had was largely related to that of muscle, so the impact on skeletal muscle. But with this slide, what I wanted to just point out to you is that SIRT1 has many roles on many different tissues and not just skeletal muscle. And some of the ways that we've looked at it in muscle are complicated by the fact that we haven't always looked at other tissues that may be impacting directly or indirectly what's going on back to the muscle. So our interest has been really the regulation of SIRT1, SIRT2-1, which is a NAD-dependent deacetylase. So it's going to go and deacetylate a number of proteins, and there's two listed there, PGC1 alpha, which you I'm sure know is a master regulator of mitochondrial biogenesis, and then the FOXO family. And in skeletal muscle, our interest has been in sarcopenia, regulation of apoptosis, mitochondria function, autophagy, mitophagy, and then the cell cycle control pieces that would be involving in muscle stem cells. And more recently, with my collaborator, Dr. Junaid Mohammed, we've been interested in what skeletal muscle reactions are with undergoing some central nervous system disruptions like stroke as a model, and how SIRT1 might impact those kinds of issues. So just to take a moment for the ROS subproblem, a recent paper by Scott Powers summarized quite nicely some of the main sources of ROS production in skeletal muscle. This is really a very familiar story, I think, to most people. We've been sort of thinking about or concentrating more on the mitochondrial sources and the impact that excessive ROS has in mitochondria, but there's many other places that ROS can be generated from. But mitochondria, for example, are one source that can be damaged by mitochondria, but also can lipids and can protein and DNA, et cetera. So there's all these markers that we can use either as direct or indirect measures of how much production of reactive oxygen species we can have in a system. Often in hydrogen peroxide has been the marker that we've used a lot as a pseudomarker of what's going on in reactive oxygen production. But we also have to recognize that antioxidants are also regulated and generated, SOD1, SOD2, for example, and others in the system. So there's flux of reactive oxygen species going on. And the other piece of this, and we'll come back to this as well, but just having ROS in a system doesn't necessarily become a bad thing. It's just when it becomes higher, excessive, and not able to be buffered by the systems in place as antioxidants. Some classic work from Dr. Hood's lab a few years ago now has shown very clearly that with aging, we have this loss of muscle mass, sarcopenia, that we were aware of, but that is also associated with increases in ROS production, and this was in the subsarcolemma mitochondria population, as well as interfibrillar mitochondria population. But you see particularly the subsarcolemma population of mitochondria appear to be producing more reactive oxygen species in an older animal, so a 36-month-old animal, compared to a 6-month-old animal. And this has been relatively consistent across the literature, that with aging, we have an increased basal level of reactive oxygen species just being generated in muscle. In this case, it's in mitochondria, subpopulations of mitochondria. And it turned out that this increase in ROS also was associated with a lower membrane potential in mitochondria, suggesting that they're not quite as healthy in muscles of old animals as compared to the younger animals. And so this is relatively a common theme that we see across aging, that there's this increased levels of ROS chronically, basally, along the system, and therefore it has to have an impact on the situation of the tissues around it. So high levels of ROS, we know, contribute or can contribute to damage in muscle, can contribute to muscle cell death in culture, and I'll show you some of the data that we've looked at to support that. We know that it can contribute to things like apoptosis, as well as decreasing the ability to repair muscle. So the question related to SIRT1 is, can SIRT1 that has this deacetylase activity for things like PGC1 alpha, and well, I'll show you some data to suggest that it also can increase antioxidants in muscle and therefore buffer some of the ROS. Can this protein be used as a therapeutic, or does it have any important role in regulation of ROS at all in the muscle? Some of our studies have used a indirect activator of SIRT1, which is resveratrol. And we found, and others have found as well, that the activity of SIRT1 is increased with resveratrol being introduced to animals, or humans for that matter too. So resveratrol is one of the things that we've studied in that context. And this is our data from work that Mike Ryan did in my lab, where we fed animals, these are mice, and these were comparing young mice in the black bars to older mice in the gray bars, and these are comparing controlled diets. Now, what you see is that just basal levels of reactive oxygen species, at least as measured by hydrogen peroxide, is just significantly higher in the muscle of older animals. When you exercise them acutely, and this is after 3 days of isometric exercise on an Aurora system, 3 days of exercise increases the reactive oxygen species production in the muscle even more. So that's not that surprising. Exercise induces ROS. ROS can be a signaler for a number of different pathways. So that's not unexpected and really not necessarily a bad thing. But the problem becomes if you've got a high basal level of reactive oxygen species to begin with, that could potentially cause damage to the muscle, and you're increasing it even further if you don't have compensation from antioxidants. Will that induce more damage to the muscle? So does acute activity become something that one has to take special attention to? So if you have an old person who hasn't exercised for a long time, and then you just throw them in the gym and give them 3 hours of exercise, which hopefully you wouldn't do, would that be a good or bad thing? Well, it probably wouldn't be a great thing for lots of reasons, but it could induce more damage acutely. There would have to be a ramp up period until there's some compensation. But the point for this is that we're able to measure increases, higher levels of ROS in muscle that can be suppressed if we put resveratrol in their diet. And this is only a 10-day diet, and it's a very low percentage of their diet that was consisted of resveratrol, this SIRT1 activator. And what we find is that the basal levels of hydrogen peroxide are suppressed in the old muscles. In young muscles, they've really no difference after their acute exercise pieces. and exercise ROS production was significantly suppressed as well. So what this suggests is that activation of sirtuin-1 really can suppress the production or at least accumulation of hydrogen peroxide in this case. In terms of does it cause damage? Well, if we use a marker like lipid peroxidation, which we've used MDA and HAE as an indicator of oxidative damage, potentially at least to membranes, lipids, what we find is that aging does have higher levels of lipid peroxidation in muscles, and these are really whole muscle homogenates at this point. And after acute exercise, of course, now it's gone up even to higher levels, whereas resveratrol acts as a buffer and prevents much of that increase, but is not able to bring it down to that of young animals. So it is suppressing, but it's not preventing the increases basally that we just see in the muscles of older animals as compared to young animals. So what this suggests is that resveratrol does have the ability to buffer the excess ROS that's being produced, but it doesn't bring it back to the levels of young animals. And we know from other studies and some of our work and other work that high limb suspension disuse, and this is a model for disuse, can cause increases in reactive oxygen species and affect muscle and mitochondria and other things too. So we wanted to know whether resveratrol activating sirtuin-1 would in fact be a modulator of reactive oxygen species production. And what we found is that young animals, and these are comparing the control to a resveratrol-fed young, and this is a young animal that's been suspended, so high limb suspension, and this has been for 14 days, and these are in Fisher-Brown Norway rats. And that was compared to old animals. These are just control animals. Again, basal levels of hydrogen peroxide much higher than young animals. Resveratrol suppressing it some, but not hugely. Suspended animals, we see an increase in hydrogen peroxide production with suspension relative to the control animal. But again, what resveratrol does is some suppression, but it's not a miracle worker. It's not dropping reactive oxygen species down to the floor by any means. The same kinds of things that we'd see occur over with the lipid peroxidation. So we see a suppression with resveratrol, but we don't see any total elimination of it. So we see some buffering associated with that. We also see part of the reason likely is that activation of sirtuin-1 is increasing some antioxidants, and this is MNSOD increasing in control animals fed resveratrol that are old, and suspended animals, the same idea. So we've got this increase in antioxidant production in muscle, which you would think would be really important for buffering those excessive reactive oxygen species that are generated under a situation of disuse or high limb suspension, as is the case here. And this is just looking at content, whereas the one on the left is looking at activity. And we do see that the content is increasing as well. So we see that resveratrol is reducing reactive oxygen species accumulation, at least in part by having antioxidants that are increasing in the system. In terms of other antioxidants, the same kind of story occurs, and I'm not going to spend a lot of time on this, but really resveratrol is pushing some of the other antioxidants up as compared to standard diet, and these again are in the same order with aged animals and these are exercised rather than suspended, but the same idea occurs that we've got resveratrol increasing antioxidants along the way here. So we see a relatively consistent story that we're seeing an increase in antioxidants in aged muscles under numbers of conditions. These are in high limb suspended rats in this case as well, and showing much the same kind of information with suspension with resveratrol having higher antioxidants than without the resveratrol or sirtuin activated piece being there, and lower lipid oxidation and lower hydrogen peroxide levels. So the stories are fairly consistent. Now, the problem is that, and that looked all really good for a while, but the problem is that with aging, we don't see a significant impact on muscle atrophy, or specifically sarcopenia in this case. So this was a study where we fed mice starting at 18 months until they were 28 months. So 10 months of feeding of from middle age to pretty old animals, not quite frail, but getting close to that, with resveratrol. And this was the 12.5 milligrams per kilogram per day. So these animals didn't eat different from their pair-fed controls that didn't have resveratrol in them. SIRT1 activity was higher in the old animals. These are 28-month. These are 28-month that had resveratrol. These are compared to the 18-month-old animals, and this is the 6-month-old animals. So you see that the resveratrol did have an increase in SIRT1 activity relative to the aging component, and total protein for SIRT1 went up too. So it did what we had expected it to do. It also increased the MSOD activity, so the antioxidants increased. So we buffered, and I won't show you all the data, but we buffered a number of antioxidants as well. The antioxidants went up, SIRT1 activity went up, SIRT1 protein went up in these old animals. What we didn't see were the effects that we were expecting. So lipid peroxidation didn't improve with the resveratrol. Protein carbonylation, carbonose didn't improve. Hydrogen peroxide didn't, drop through the floor. It was less, but it wasn't humongously so. But the real kicker was that we really didn't see a change in the amount of muscle mass with resveratrol fed as compared to those without resveratrol, meaning that we still saw sarcopenia occurring. And despite the fact that we buffered some ROS, presumably, we've increased SIRT1 activity, and the things that we found acutely it worked on, it really didn't affect the bottom line, which was trying to prevent atrophy in these old animals. So that was a little bit disappointing in the sense that in terms of a treatment or something that maybe activate the system with the SIRT1, just not panning out completely like we would have anticipated it to do. It doesn't mean it doesn't have acute effects, though. So we were also interested in muscle repair and myogenesis and trying to look at some of the acute effects of resveratrol. Now, back to the ROS story very quickly. What we know, and this is a nice paper that was just published in 2020 that is a review showing, summarizing at least, that in muscle cells that are required for regeneration, be muscle stem cells or satellite cells, there's generally a level of ROS that's pretty low. And as we move into from stem cells to division and then finally to fusion into differentiating fibers where the repair pieces would happen, ROS increases. But it turns out that if you have higher levels of ROS at this level, when you are dividing satellite cells, that there can be a real problem with that. And this is a cartoon example of what a muscle would look like if you had a conditions of exercise or caloric restriction or something, and if you're going to damage and repair the muscle, you've got a pretty good turnover of activation of satellite cells, and they do what they're supposed to do in terms of muscle repair. If you've got a situation where ROS is higher, where obesity induces ROS, where muscle wasting is in place, again, ROS being higher, other things may be going on, but that's certainly part of it. ROS being higher, produced at a higher level, actually interferes with the proliferation of those muscle stem cells. And if you don't have as many muscle stem cells, muscle repair is going to be worse. So the pieces that ROS is involved with muscle repair is complex it needs to be low in the beginning during the division, but it can be tolerated at higher levels in for example, in differentiated cells. And we saw this in some of our own studies where we were artificially looking at very modest levels of hydrogen peroxide, 100 micromolar, and comparing that to a hammer, which is a 1 millimolar concentration of hydrogen peroxide, and looked at what differentiated myotubes would look like. And as you might expect, if you're incubating cells, in this case, myotubes MB is myoblasts, and then myotubes is the MTs. We really did both of those kinds of things. But what essentially you find is that the higher the level and the longer duration of exposure to hydrogen peroxide or ROS results in a bigger loss. So the cells start to die, is what that really is saying. And part of the way that they die is through apoptosis. So this is a treatment with our hammer with 1 millimolar of H2O2. And you see that myoblasts undergo a lot of apoptosis, that at least is labeled from tunnel labeling. Myotubes do, but they're much more resistant to hydrogen peroxide. Again, kind of along with the idea that differentiated cells are easily or more easily able to handle the hydrogen peroxide or ROS pieces. This just gives you some apoptotic indications, whereas myoblasts have a much higher death rate than myotubes for a given concentration of H2O2. But the important thing is that the hydrogen peroxide or the ROS is going to, in excessive levels, cause death. And that's really what we see with aging. If we provide resveratrol to our cultures, either myoblasts or myotubes, in this case, this is looking at our 100 micromolar or the black bars or the 1 millimolar concentrations of hydrogen peroxide. And we give resveratrol to the cultures, and these are control levels of 0 up to 50 micromole of resveratrol, and then look at some apoptotic markers, and these are after incubation resveratrol or no resveratrol, depending on the concentration, for 24 or 48 hours. What you see is the higher levels of concentration of resveratrol reduce the activity of the apoptotic markers, in this case, caspase 9, caspase 3, the same kinds of things after 24 or 48 hours, and even caspase 8, which is really more of a TNF alpha or non-mitochondrial marker of apoptosis. But the message is still the same, is that resveratrol appears to be protective against death to these cells. And in this case, it was myoblasts, but we did much of the same thing with myotubes. In the case of myoblasts as well, what we found is that resveratrol with higher concentrations actually reversed the loss that hydrogen peroxide had in SIRT2 So the high levels of ROS reduces the SIRT1 protein levels in the cells, and resveratrol reverses that. So it increases it back, essentially back to control levels, as we saw there. So we do see some protection of resveratrol against ROS in these muscle stem cells or muscle stem cell markers. These are C2, C12 myoblasts, but as a marker of what a muscle stem cell would look like. And then the implications are more towards a regeneration after that. And to ensure that we weren't being fooled by the C2, C12 cells, we isolated muscle stem cells from our mice. This happened to be the SYN4 muscle stem cell population. Other people use other markers. This is a marker that Thomas Rando had used a few years ago, but it identifies a population of muscle stem cells. And then we looked at the SIRT1 activity levels in just control cells. We then added a SIRT2 inhibitor, an NAD inhibitor, which then would reduce activity of SIRT2 because SIRT1 is NAD dependent. It needs NAD in order to be able to activate it. So it wasn't surprising then if we inhibited NAD that SIRT1 activity is reduced. Resveratrol reversed that, brought activity back up as we would have expected. It buffers the loss that NAD loss would do if we give resveratrol to the system. H2O2, of course, reduces SIRT1 activity, as we saw previously in other ways. And if we can reduce it even further, if we lower NAD and we also have ROS produced, because that's going to cause death and other things that we've talked about, that can be reversed by resveratrol, and resveratrol at least partially rescues the whole piece, even if NAD isn't sufficient. So SIRT1 activity can be elevated from resveratrol. SIRT1 protein can be elevated as well. So we certainly have the pieces in place to think that resveratrol is going to be an important activator of SIRT1, and therefore in these muscle cells that were derived from skeletal muscle, not just from the C2C12 line, we see the same kind of story. So we're fairly certain then that this is actually giving us the data that we think that we were going to see from the previous work. So now, does it make a difference in muscle repair? And that's really the next really important question. So in terms of what it does in the system, if we looked at, and this is back to our C2C12 model, if we just look at myosin heavy chain, which is the red marker, and then begin to look at the differentiation components, we see adequate differentiation with resveratrol, but we see that if we inhibit SIRT1 activity, then differentiation, myotube formation is inhibited, which would imply that if you inhibit SIRT1 in a muscle repair situation, that the repair is going to be poor if this continues to hold through. So the cell culture data suggested that inhibition of SIRT1 activity is going to be reducing myogenesis, and then when we moved it up, it enhanced it. We also developed mice that were a knockout for SIRT1. This is a global knockout in this case, and these are flox mice. And what we found is that the muscles were not hugely different with the knockout there. The fibers, in terms of, are actually a little bit smaller, and I'll show you some of that in a second. But if you inject the muscle, and this is a tibialis anterior, with a cardiotoxin to damage the muscle, you would expect the muscle to be injured and then undergo repair, and that's from a satellite cell, muscle stem cell derived mechanism. You see pretty good recovery. This is 8 days post. What you see on the knockout side, the knockout animals had significantly poor recovery of the muscle, suggesting, as we would have thought from the previous slide, that the maturation of myotubes after they have been formed, or the activation, differentiation muscle stem cells, has not been processed in the same fashion and at least the same rate as when SIRT1 is present. If you wait long enough, and this is 24 days after the cardiotoxin injection, repair occurs pretty well. The muscle doesn't look really too bad in the knockout situation. So what this tells us is that SIRT1 is not essential for muscle repair, but it does enhance it because these fibers are still smaller and we still have more fibrotic areas through there. Looking at the proliferation pieces, these are in young animals. What we found is that when we overexpressed or had an overexpressor animal that was damaged by CTX, and then put bromodeoxyuridine in as a marker for cells that proliferate, it's a thymidine analog, so it would be picked up each time that the cell divided. We had much more division, much more proliferation when SIRT1 was overexpressed, and certainly compared to that when SIRT1 was lost. So it didn't eliminate it completely at all, but it certainly was markedly reduced, and compared that to either the control or wild type, really two controls in that case. So this suggested that there could be a role for SIRT1 in recovery, or in divisions, at least, of satellite cells, proliferation of satellite cells. Muscle-specific knockout animals, again, the muscles aren't hugely different, but those fibers are smaller in a basal level if you just chronically knock SIRT1 out of the muscles as compared to wild type. There's a slight shift to the left in the sizes of fibers, with the knockouts being, again, slightly smaller in all cases. But look what happens when you damage the muscle and then you allow it to recover. So CTX damage, 24 days later, you do not see the recovery of the muscle fiber sizes in a knockout animal back to the place where the control would be, that had been damaged, but it had repaired. And this is the evidence for that in terms of fiber sizes, where you see a significant shift to the left, which are the red knockout fiber sizes, as compared to the control fiber sizes that had been damaged and then allowed to recover. So this, again, supports the idea that SIRT1 may be important in that differentiation, that anabolic sort of side of satellite cells coming together and then incorporating themselves into existing muscle fibers, repairing, and so on. So it appears that the loss did impair SIRT1 activity. So as we went through, we then wanted to ask the question, if we lose SIRT1 in a muscle, is it important to lose it in the muscle itself, the muscle myonuclei, non-dividing myonuclei? The satellite cells might also be the other place. So we developed muscle-specific knockout, which is this animal, and a muscle stem cell specific, so a satellite cell based on PAX7 labeling knockout mouse. And then we allowed some of the mice to age up to 80 weeks, which is not a really super old animal, but it's an older animal, and compared the responses to muscle repair to the young animals. So until this point, the things look relatively clear, but it gets a little more mucky now. We induced cardiotoxin injury and then had 21 days of repair to the muscle. These are just some Western blots comparing PBS sham injected animals and cardiotoxin injected animals. This is an overexpressor animal. This is a global overexpressor where we just had more SIRT1 protein, and these are SIRT1 blots here. This is a muscle knockout of SIRT1, and this is a satellite cell knockout. Now, one of the things to note with the satellite cell knockout is that although SIRT1 was knocked out in the satellite cells, the PAX7 cells, it was not knocked out in the myonuclei of those cells. So it creates a little bit more of a question in some of the pieces in terms of, was one able to compensate for the other? Now, let me just show you very quickly a couple of pieces here, which again suggest that there's a lot of question marks in terms of how SIRT1 might be regulating the myonuclei versus the satellite cell population of cells. So this is SIRT1 expression after 21 days after injury in an old overexpressor muscle, as compared to, and this is a control one that had just been injected with PBS, and then a CTX injected. Looks like there's a compensation with injury and repair. So SIRT1 is really not different from the control animal side of things. But this is all, both of these are larger than the amounts of any of the other muscles. So overexpression works. It has higher levels of protein, and the CTX didn't bother that at all. It didn't really suppress that. Satellite cells, as you might expect, had very low levels of SIRT2 in that, but they still had some because they were in the myonuclei. CTX injected repaired muscle seemed to compensate and increase that a little bit, but they were still lower than what was going on in the control muscles. So what this suggested was that although SIRT1 was greater in the overexpressor muscle, it really didn't increase after the injury repair cycle at all. It was interesting, but not completely clear on what this all means, but P53 was also elevated in a couple of these samples, and it seemed like there was an interaction between P53 and SIRT1 in the regulation of what was happening in the muscle, in the sense that the ones that had higher P53 seemed to have a better response, interestingly. But what was really intriguing and not necessarily clear was that muscles that had a satellite cell knockout of SIRT1 had lower levels of force production. So the muscles were weaker. They weren't necessarily hugely smaller than the other groups, but they were weaker. Whereas there really wasn't any major things that were going on in terms of force production, whether you overexpressed SIRT1 in an old animal, or you had a muscle knockout in an old animal, or you had it recover afterwards. So these are the conditions before the damage occurred. This is a PBS injected and CTX injected. And really, you see it recovers adequately after the 21 days. So, but what this suggested was that the muscle stem cell loss of SIRT1, but not myofiber loss of SIRT1, somehow reduced the ability to generate force. So muscle SIRT1 was not essential for muscle force recovery or production of force after repair, because overexpression of SIRT1 did not improve force with injury. So really what you wonder, though, is if that's a neural aspect going on. Here's the fiber data showing that even though force is lower in the satellite cell knockout animals, fiber area was not. It really wasn't impaired in either the injury recovery or as compared to the muscle knockout or even the overexpressor. It really didn't change very much. So while the fiber was able to repair okay, at least over this period of time, it did not appear that overexpression of SIRT1 or loss of SIRT1 really impacted the structure of that repair in these particular mice. So the disconnect between force and fiber size suggests that maybe there's something going on at the neural aspect, either neural, the neuromuscular junction, or some aspect of that wasn't muscle specific could be part of the issue. In terms of muscle proliferation of satellite cells, we didn't see things that we probably completely expected based on our previous data. So the overexpressing animal didn't have more BRDU or proliferation of satellite cells in response to injury as compared to the wild type. And in fact, almost any manipulation of SIRT1, whether it's overexpression, a knockout in the muscle, or knockout of the satellite cells, seemed to depress the activation of the proliferation in this particular study. So all manipulation seemed to reduce proliferation. But the question would be, does that mean that it's less injury? Maybe these animals were more resistant to the CTX injury if they were overexpressed or something else was going on. Or does it mean that so they didn't need to repair, therefore they didn't need to have the increases of satellite cell activation? Or was there something else going on? To look at the stem cells themselves, we isolated stem cells from the mouse muscles, again, not C2, C12, but from the mouse muscles, and then looked at proliferation in response to an EDU assay. And at least in vitro, the muscle knockout, or the stem cells isolated from muscle knockout, actually proliferated more than any other combination. So satellite cell knockout had some proliferation, but it was lower than the muscle knockout. And what this suggests is that there's some kind of crosstalk now between the muscle that has no SIRT1 and the satellite cell that's sitting out there that could still have SIRT1, but be able to cause the division in that. So this greater proliferation in this particular population was really kind of intriguing, but more of a mystery than anything. And it does suggest that maybe there is some crosstalk between the muscle compartment and the satellite cell compartment, at least in this element. But yet overexpression of SIRT1 or knockout of the muscle stem cell SIRT1 didn't really appear to affect or at least change proliferation very much, which suggests there's really not much crosstalk in those particular models. I think time is moving on, so I just wanted to touch a couple of other pieces related to how SIRT1 is affecting some other pieces, including mitochondria. What we saw in a study where we fed mice resveratrol, then isolated mitochondria, that the mitochondria permeability pore was more stable. It didn't open in response to a raw signal to the same extent that wild type or control mice fed non-resveratrol diets did. So that was suggesting that, well, maybe resveratrol could have a function on the mitochondria, not necessarily an ATP regulating or a oxidative phosphorylation mechanism, but there could be other signaling through that. When we looked at complex 1 activity in young mice that were given resveratrol, or no, these are not resveratrol, these are the SIRT1 overexpressors and the SIRT1 knockout animals. What we found is that complex 1 actually tended to be higher in SIRT1 overexpressor muscles. And when we damaged the muscle with CTX, they had an even greater increase in complex 1 activity. So what this was suggesting was that SIRT1 overexpression could in fact potentially affect mitochondria function, at least from complex 1 markers. But will that have any translation to repair? These are just data showing that not only does complex 1 change, but in the muscles that are muscles of in a different study that underwent hind limb suspension, and then the animals were allowed to walk around their cages and recover, that animals fed resveratrol had higher levels of PGC1 alpha, which would suggest that mitochondrial biogenesis could potentially be higher in those muscles after the atrophy and then recovery from atrophy situation. So this did point to a potential role that there could be an anabolic piece to this, at least a piece where mitochondria amounts could be higher. And therefore, we are wondering if the mitochondria itself had maybe changed as a result of the damage and recovery and then having SIRT1 present or absent. So what we did was to take our mice that were either wild type, and these are the wild type older mice of 80 weeks, overexpressing mice, the muscle knockout mice of 80 weeks, and then the satellite cell knockout for 80 weeks. What we found is that the mitochondria that we isolated from skeletal muscles of these animals after 21 days of recovery from the CTX damage, and then used a FACS analysis to quantitatively look at the size of those mitochondria using forward scatter and side scatter measurements. What we found largely was that the muscle mitochondria were just larger in muscle or mitochondria from muscles that either overexpressed, knocked out, or had satellite cell knockout. And this is a subsarcolemma mitochondria population specifically. And so just sort of any manipulation looked like the subsarcolemma mitochondria were affected by getting larger. Now, what we didn't do was to look at whether the fusion or fission proteins were being altered as a result of that. The interfibrillar mitochondria, though, showed the same kind of trend where they were larger in terms of the forward scatter size from the FACS analysis as well. and their membrane potential was a little bit lower. And I did mention, and probably should, that the mitochondria membrane potential appeared to be, though, back to control levels in animals that had knockouts of SIRT1, either in the satellite cell or muscle, in that wild type or overexpressing SIRT1 animals had lower membrane potentials for the subsarcolemma mitochondria based on the dye assay. So this looked like it was sort of normalizing the membrane potential mitochondria by having a knockout, not an overexpressing piece. Complex activity, really not a lot of excitement here at all. There's a little bit of complex 3 activity differences, all being lower in the overexpress or knockout as compared to wild type animals, but really in all, not really much going on. No clear deficit or advantage of maximal mitochondrial activity when SIRT1 is active or present in these particular mice. Fatigue-wise, there's a little bit of suggestion that fatigability was improved, either with a muscle knockout or an overexpressor, but not really wildly changing, and really not a good explanation based on the muscle of the mitochondria. So I'm not really convinced that it's yet a mitochondria function, but it could be that we haven't ruled out. Very briefly, I just want to mention some work that my collaborator, Junaid Mohammed, has looked at as a competitor to SIRT1, and that is PARP1. And PARP1 is an activator, well, it's an NAD activated protein involved in DNA damage repair. And it turns out when PARP1 is high, then SIRT1 gets inhibited. And in the sense of time, I think I'm just going to skip very quickly through this, but showing that if you have high levels of ROS and you inhibit PARP1, then you can recover some of the SIRT1 protein levels and activity as well. So if you inhibit PARP1, activity of SIRT1 is up, and this is based on acetylation of lysine on PGC1 alpha. So less protein here in acetylation means it's been deacetylated, which means that there's more activity. So that means that the SIRT1 activity is higher when PARP1 is inhibited. And that makes some sense because they're both SIRT1 and PARP1 are dependent upon having NAD available to them. And if PARP1 is taking all the NAD, there's less available for SIRT1. And that's essentially what this next piece shows. And I think my time is getting away. So I'm just going to skip over this very quickly. Dr. Mohammed inhibited PARP a couple of different ways, either by siRNA or by JP34 in mice or in cells, and showed much the same thing, that if you inhibited PARP1, SIRT1 activity is higher, and then some of the other activities are improved. Mitochondria function and mitochondria genes are actually increased, and muscle fatigue is improved when PARP1 is inhibited in these animals. So these are not overexpressing animals, these are just older mice. Just sort of following on that story, some recent work that is continuing in Dr. Mohammed's lab includes the stroke model, the middle cerebral artery occlusion model. So it induces a central damage, but then looking at skeletal muscle, and this is tibialis anterior on the side that undergoes paralysis, you see a loss of SIRT1 in these stroked animals and an increase in acetylated lysine, which means SIRT1 activity is lower. So there's certainly indications that there is this neural component. It turns out that if you inhibit PARP1, then you can also find improvements in things like the atrogen population. So this is for American folks, ZNF216, and for Canadian folks, it's ZNF216, which is a 26S proteasome. But atrogen MERF and the 26S proteasome are all really active in stroked muscles which are undergoing atrophy. And if you inhibit PARP1 through siRNA, then you reduce the 26S proteasome activity, suggesting that PARP1 is upstream of that. But if you give them resveratrol, all the atrogens are going down, including the proteasome. So again, suggesting that there's this competition, but resveratrol appears to be further upstream than that. Given the time, I think I'm just going to move on to the last piece, which is some of the data that we collected ever so superficially, perhaps, in humans, trying to make that application. Does SIRT1 activity actually make a difference in muscle adaptation and aging? And in this study, we collaborated with a number of clinical faculty. We obtained biopsies from the vastus lateralis. We exercised them. And then we had a double-blind study where we gave subjects 500 milligrams of resveratrol twice a day for 12 weeks and rebiopsied them and redid all the data. What we found, just very quickly, is that our exercise was pretty modest. So VO2 max in these older people, and the average age was 72 in both groups. So they're not really old, but they're getting older. Didn't change very much, but when we gave them resveratrol, VO2 max increased, and when expressed per body mass index, then that increases as well. There was some suggestion that mitochondria volume maybe increased a little bit. They looked a little bit more healthy, but we would have to really dig into this much more because we can't rule out sampling bias or error with our muscle biopsies. But it was suggesting that we're moving towards a mitochondria piece, which we know is impaired with aging, with atrophy. So just putting this all together very quickly in the last minute, our working model is still a working model. If we start out with a muscle that's undergoing atrophy, we know with aging, we know with disuse, we have this big ROS signal. The ROS is going to be magnified with aging. It's going to be in high levels affecting mitochondrial function. We know that it inhibits satellite cell proliferation, so muscle repair is reduced. We know with aging and ROS that SIRT1 is going to be inhibited. If SIRT1 is inhibited, then atrogens are going to be allowed to increase because SIRT1 does inhibit the atrogen genes through this. If atrogens are high, muscle repair is reduced. Atrophy is going to be part of the product. ROS, as we know now, increases PARP1, as does aging. And this competes for NAD, which means there's not as much NAD available for SIRT1, which then goes down this other pathway to even make it worse. Exercise, and we haven't had much time to talk about this, is going to activate SIRT1. Resveratrol certainly does. And because if we're increasing SIRT1, we're going to be blocking the atrogen effects. We're going to be blocking effects of ROS. Once we activate SIRT1 with NAD and get PARP1 lower, we've got PGC1 alpha increasing, which means mitochondria biogenesis can presumably increase. And by having more mitochondria, then we're going to be inhibiting the inhibiting part of repair, potentially improving muscle repair. So this is part of the question that we're trying to address at the moment, is what role mitochondrial function has in muscle repair in this sort of sense. Myogenesis is a little less clear, and our knockout animals or overexpressing animals didn't really help solve our questions. It looks like SIRT1 does have a role in proliferation, certainly in the in vitro studies. It's just less clear in our in vivo animal model studies, but those are certainly complicated animal models. And there's certainly possibilities that compensation for other tissues that we talked about in the very first slide, because SIRT1 has roles in lots of other tissues other than skeletal muscle. Let me just really quickly just acknowledge the folks that had involvements in this study. Dr. Muhammad, a previous graduate students, Matt Myers, Mike, Jana Carell, current students in the lab, Chris and Hector, and Peter is working with Dr. Muhammad on the stroke and atrophy side, our clinical collaborators, and our funding, which is always important for doing any studies. So I will stop there, and thank you very much for your attention, and happy to answer any questions.